Okie dokie. So I prefer to use the black screen. Quick note regarding part of what we're about to talk about, but more with relation to the distal convoluted tubule and collecting duct and the renin-angiotensin-aldosterone system. I made a podcast episode that talks about that over the weekend. It's up now on the YouTube, or not YouTube, excuse me, the Spotify thingy. So if you just go search Christina Physiology Corner with Professor Howard, that's my podcast. It's on Spotify. You can get to it from there. And I just uploaded one that specifically talks about the relationship between left-sided systolic heart failure and the renin-angiotensin-aldosterone system, which is helpful because it gives you a recap of two systems that you've learned and discusses how they relate to each other on terms of heart failure. So it's both interesting and also helpful for review. So go check that out as well. I already sent the link to it to some people, so FYI. Okay, so this video is going to be about the nephron loop and countercurrent exchange / multiplication. And I'll explain why I'm using both of those terms in just a minute. Crud. Okay. Yes, I would like to enter tablet mode. Thank you for asking. Actually, I don't. Never mind. Okay, so let's give this sucker a title. Not quite that big. All right, so countercurrent exchange / multiplier in renal physiology. So the reason I'm using exchange here is because within the nephron loop, it's still a tube and it still has filtrate in it that's on its way to being urine, but isn't quite urine yet. And in order to get it closer to actually being finished urine, meaning we've extracted all the stuff we want out of it, we've put all the stuff we don't want into it, and it's now actually pee as opposed to an intermediary, that necessitates exchange between what's inside of the tube and the interstitial fluid and ultimately the capillaries, in this case of the Vasa recta. It's called the multiplier because of how it functions. So multiplier is talking about the ability for transport within one tube to multiply the maximum potential reabsorption in another tube. So basically, it supercharges H2O reabsorption, because obviously, since we filter so many times the total volume of our blood every day, if we didn't get water back out of that filtrate, we would just simply pee ourselves to death, which sounds miserable. So instead, we do all this fancy stuff by manipulating osmosis to get most of that water back, and that helps us keep our blood volume up. And the renin-angiotensin-aldosterone system is part of that, but the nephron loop is another part. The other thing I want to address before we talk about this is countercurrent. So this refers to flow in opposite directions. So this isn't the only example of countercurrent exchange in biology. There are actually many others in lots of different organisms too. So any animal with a kidney is going to have some kind of nephron loop. So that's one place countercurrent exchange is useful. But it's also useful in fish gills to, put them in contact with water and oxygenate the blood in the vasculature of the gills and supercharge oxygen reabsorption for fish, anything else with gills. And then in animals that live in extreme environments that are very, very cold, you don't want to lose heat through your feet, which are in contact with the cold ground. So there's actually a countercurrent multiplier in the blood vessels of those animals, similar to the countercurrent exchanger in the spermatic cord between the pampiniform plexus and the testicular artery, where the veins serve as a heat sink for the arteries so that blood is warmed back up on its way back up the feet. So if you've ever looked at wolves or penguins or polar bears and wondered, like, why don't their feet just freeze off? Countercurrent multiplication is the key to that. So lots of cool applications. Here's the one that we use for our urinary system. So I don't think I'm going to have enough room to draw all the stuff that I want to draw big enough without erasing this. So I'm going to erase it. And then we're just going to rewrite. So I already have a YouTube video about this on the short videos channel, but I don't think it hurts to have an extra one because every time I explain things, it's a little bit different. And so there's going to be 2 videos with the nephron loop on this thing. That's fine. Okay, so let me just really quickly draw our nephron. And I'm going to kind of de-emphasize the parts I'm not going to talk about, but it's still worth putting them there just so that you have an idea of the context for all this stuff. Okay, so as you can probably tell, that's my glomerulus. And then we have the capsule, which gives rise to our proximal convoluted tubule, which I'm not going to make all that long. And then that gives rise to the thin loop. And then after the loop, which I'm going to draw inset in a box, is our DCT, distal convoluted tubule. And then that plugs into a collecting duct, which also receives input from lots of other distal convoluted tubules. So this kind of gets at the question from the person who is like, it's hard for me to understand the collecting duct and which things it's attached to. Totally. So let's provide a little bit of that context here, and then we can sort of enhance it later when we have a better understanding of our loop. Okay, so I'm going to do this in an inset just so that it's separated from the other contents, but also because I need to draw it a little bit bigger. I'm just going to erase some of this for maximum bigness of my little box. What if I do it from the bottom up? That should give me more height. Yeah. Okay. So I'll label all these things as I customarily do as well, so you won't have to like look back and be like, which tube was that? I don't remember. Okay, so in our nephron loop, which is also called the loop of Henle, so just so you know, those 2 terms are interchangeable. As usual, nephron loop describes its anatomical context. Henle refers to a guy that was like, hey, what's this loop thingy do? Oh, cool, I made a discovery. I'm gonna name it after me because everybody needs to know that I figured out the pee-pee loop. That's how lots of things in A&P are named. But if you discover something, you get to name whatever you want. So usually anatomists name stuff after themselves, but other kinds of biologists are more fun. Like somebody discovered a new bumblebee a couple years ago and named it after Beyonce. True story. So if you discover something, it's up to you. Okay, so coming off of our proximal convoluted tubule, we know that we've already done some tubular reabsorption and secretion. But then we have this long loop that dips way down into the renal medulla. So the PCT, the DCT, and the renal corpuscle, which includes the glomerulus, those things are all located in the renal cortex, so the tissue on the edge. And then the renal medulla is organized into sort of cone-shaped regions that are called renal pyramids. And that functions as the medulla. So the medulla of a kidney has a lot of powerful features. And one of those features is it creates and maintains this really steep concentration gradient where at the top of the renal pyramid, so the side closest to the cortex, the osmolarity is almost the same as the interstitial fluid and the blood, which is about 300 milliosmoles. I'll label that. But the deeper you go into a renal pyramid, the saltier and saltier and saltier it gets. And from our discussions of colloid osmotic pressure, you'll probably be able to connect the dot that higher osmolarity equals more power to attract water via osmosis. And so you can fiddle around with gradients to attract water strategically. And we've already looked at a couple places where that's true. Here's another one. And then in addition to the power of the loop and the salt that it reclaims, and I'll talk about that in just a minute. Actually, let's do this another way. In addition to that, and this gets at a question that was asked by a student today via email, and I answered her, but I also made a note to myself to talk about it today here. And we're just going to make this artificially very fat because I need room to draw stuff. Okay. So another thing is that urea, which is a byproduct of protein catabolism, so the breakdown of proteins, that substance, which is nitrogenous, is a waste, which is why pee smells like pee. You know, there's that very specific pee smell. But in addition to that, we also kind of save it in our renal medulla along with some sodium chloride to make it extra high osmolarity to power this gradient. Okay, so let's begin. First things first, this is our nephron loop, and I'm going to just say this is so thin describes its epithelial characteristics. This is one of the few places in the renal tubule where we get simple squamous for a moment to facilitate exchange. And then descending refers to the direction of the filtrate flow within it. Okay, and then our thick. Ascending limb. Ascending refers to the direction of the filtrate, and then thick refers to the epithelial characteristics. So we go from being thin and descending to being thick, and that includes a transition back to simple cuboidal epithelium. So that's the loop. I also want you to know that I've made these two things artificially far apart, but in reality, they're right up against each other. That's one of the requirements for countercurrent multiplication. But if I draw it that way, I won't have room to draw all the other crap I need to draw. So that's why I'm kind of faking it. Okay, so now we have this set up. So what we're going to see is that as the filtrate moves through, and I'm going to draw some little arrows for this just to give us a visual reminder of flow direction, because the countercurrentness of it is an important feature. So countercurrent means opposite flow directions, and as you can see over on the left here, we have arrows going down, over on the right, arrows going up. So that's our countercurrent component. Now the magic of this thing is that there is a permeability difference and a transport difference between this thin part and this thick part. So I'm actually going to start with the thick part. I've overused this pink color. Let's make it another color. How about orange? So over here, I'm going to draw them on both sides, but only illustrate their function in a couple places just to save room. But all of the orange thingies are the same thing. And these are pumps that selectively move sodium chloride. I'll just draw some more. They selectively move sodium chloride out of our tubule via pumping. So that implies ATP use, and this is using ATP. So I'm just going to write an ATP in each one of these, not because this is ATP, it's not, it's a pump, but rather to indicate that it's using ATP, making this an energetically costly active transport situation. And then we're going to just show what this guy does, which is spends ATP to pump sodium and chloride out into the interstitial space. So all of the ones on the right are doing the same thing, but I'm just not going to draw it. And so once we have that happen, of course, the extracellular fluid is mostly water. And sodium chloride, which is literally the same as table salt, it's an ionic compound. So the minute it hits more water, it's going to dissociate again. So what we have is a situation where we've now got a bunch of sodium and chloride ions floating in this solution. The other component of this magical loop is permeability differences. So if this fluid is full of sodium chloride, and you know that the extracellular fluid or interstitial fluid here is mostly water, that might beg the question, okay, so if we've got all this salt in here, why isn't water osmotically moving into the thick ascending limb? And that would be a very astute question. The answer is, it's not permeable to water. So the cell junctions that join the adjacent cells together include things like tight junctions that form a zona occludens, which is a fancy way to say the occluded or blocked area. And that is a way to ensure water tightness. So even if water wanted to go in there, and it does, it can't because it's blocked. By contrast, the thin limb is very permeable to water, but not permeable to salt. And then this one, of course, because it's being transported, is. So what you have is two tubes where the flow is going in opposite directions, and they have literally the opposite permeability. So another component, of course, and we'll talk a little bit more about urea later, but for now, I think I've said about all you need to know about it. The DCT and the collecting duct are both places where urea can be moved out into the interstitial space. So a lot of the urea that we create in the course of protein catabolism does enter our urine and gives pee that very specific pee smell. Like if you're walking around and you smell pee, it's unmistakable. You're like, that is pee. It smells like pee. And one of the reasons for that is urea. It's volatile and odorous. But we do save some urea because, like in other places in the body where you have like a waste product, for example, our bodies are super duper good at being like, yes, that is a waste, but I can do some chemistry magic to turn it into something useful. So one example of that would be the use of heme breakdown products in bile. And another example of that would be, hey, we're making all this urea, so it should make itself useful and help us maintain an osmolarity gradient. So the further you go down in the medulla, due to the collecting duct going all the way to the bottom, the more urea you have. So what I'm going to do is just make a little key up here that just gives us this reminder that brown dots are urea. So you'll notice that there's a lot more at the bottom, and that's true in the kidney too. If you measure the amount of urea at the tip of a renal pyramid, it's a lot more concentrated down there than at the top. So if we know that the filtrate is largely water because it's filtered out of our blood plasma, and plasma is mostly water, then it stands to reason that the fluid descending in the descending limb is going to be still pretty rich with water. Yes, we got back some of it in the PCT, but not nearly enough. So fortunately, since we created this handy dandy gradient, let me draw some sodiums and chlorides outside the other side as well, because they do leak over from other nearby nephron loops. Because really these are all kind of smashed in next to each other. Why do I keep trying to draw chloride in green? Bad, stop. So if I'm water and I'm inside of the loop and I'm flowing downwards, because this fluid's always moving, I'm going to be like, wow, so there's so many solutes out there. I just really want to go out there. And so it does. Oops. Let me pick a different color for water. How about dark blue? There we go. Okay, so starting at the top, we're going to have water leaving. And it's doing that because, of course, we've set up this very handy urea salt gradient that is very enticing to water.
I hate to interrupt, but
Go
I just
ahead.
have a quick question.
Yeah, go for it.
The space outside of the nephron loop, would we consider that the peritubular fluid?
Yes. So any fluid in the kidney that is in between tubes, and that includes the PCT and DCT in the cortex, but also the various tubes that are present in the renal medulla, that's going to be peritubular fluid regardless, because peri just means around and tubular refers to those tubes. So yeah, it's all peritubular fluid, but what it's doing or what's going on with it is different depending on where you're at in our handy dandy nephron. Okay, one moment. Tortoise is doing his customary loudly trying to dig while I'm talking. So I'm going to let him do that in the couch because that is far less noisy. Okay, buddy, there you go. Burrow to your heart's content. Okay, great question. So here's the deal. And I'm not going to draw this over the top of everything because it would just make this drawing even busier than it already is. But I will mention it verbally and then we can add it in kind of at the last. The structure called the Vasa recta is some specialized peritubular capillaries that are specifically around the nephron loop, and also they're kind of arranged in a ladder shape. So they're also loopish, and there's little rungs of capillary going across. And so sorry. So what happens is that water diffuses out, and then it goes right into the peritubular capillaries of the vasa recta. So we're really efficiently moving water out of our blood, or excuse me, out of our filtrate and back into our blood where we need it. So I'll draw that over the top at the end, but not quite yet. So because of all this water loss, by the time the filtrate gets to the bottom of the loop, it's undergone a profound osmolarity change from where it started at the top. So now I'm going to add that in. Let's see, what color do I want to make that be? Magenta. So up here, it's about 300 milliosmoles, which is also the appropriate osmolarity for our blood to be the majority of the time. So not too salty, not completely dilute, middling. And quick reminder, milliosmol is 1,000th of an osmol, and the osmolarity of a solution is used to talk about the total solute richness of a solution. So it doesn't differentiate between solutes. All it cares about is how much stuff is in this solution that is not water. That's it. So that's what milliosmoles are expressing. And as you can see, we've got quite a bit of stuff that's not water in this solution. So due to our gradient here, as you go further and further into the medulla, the osmolarity of the peritubular fluid increases. So then by the time you get to here, it's about 600 milliosmoles. Let's make that a little bit thicker so we can see it. And then by the time you get down here, it's about 900 milliosmoles. And then by the time you get all the way down to the tip of the kidney, we're up to about 1,200 milliosmoles. So the strength of the water attracting power of the peritubular fluid increases as you move down the renal pyramid. And so what that means is, even though water is leaving at the top, and that creates a situation where the fluid inside the tubule is increasingly trying to equilibrate with whatever's outside, because remember, that's how osmosis and diffusion work. It wants to get to a point where there's about the same amount of things, water in the case of osmosis, same amount of stuff on each side of the membrane. So this is where the countercurrentness of this comes in. Of course, if this was a static fluid, if nothing was moving, we would expect water to leave the tube until it had equilibrated with the interstitial peritubular fluid, and then the net osmosis would stop because equilibrium had been reached. But we don't want to reach equilibrium. We want to keep this thing moving so we get this very concentrated solution at the end. Because what happens is, due to this osmolarity gradient in the medulla, by the time filtrate gets to the bottom, it's also at about 1,200 milliosmoles. So that's crazy, crazy higher concentration than it was when it entered at only about 300. So why it doesn't reach equilibrium and stay there is because this fluid is moving but the gradient remains the same. So we're always pumping sodium chloride into the peritubular fluid, and we're always putting urea out into the peritubular fluid in the renal medulla, and it concentrates at the bottom because of the collecting duct. So this 300 to 1200 milliosmol gradient that exists in the renal medulla, that is constantly maintained by the action of the kidneys. So the reason it doesn't equilibrate is because while that gradient is static, we have physical movement of the tubular fluid through the tube, so that, if this fluid is here and it exchanges enough water to reach equilibrium, in the next moment, it will move further down the tube, and now it's no longer at equilibrium because the equilibrium value has changed. And so it'll diffuse and osmose to reach that new equilibrium and then move down the tube to the next saltier area. So the bulk flow keeps osmosis happening and prevents equilibrium from being reached. So you need a 300 to 1200 milliosmol concentration gradient present in the renal medulla to make that happen. Okay. So now we got to consider what happens to the fluid when it goes back up. So if I have less water and I have a very, very salty filtrate by the bottom, as the filtrate ascends, due to the pumping of sodium chloride that set up this gradient to begin with, those are always on. We're always spending ATP to do this, so it's not passive. But that means that now we have this very salty fluid that we're going to start wringing salt out of on the way back up. So that's going to desaltify. What's the word? My brain is broken today. Desalinate. There we go. I know words and they mean stuff. Desalinate the water that's coming up so that when you get back up to the top of the loop and the filtrate is entering the DCT, its osmolarity is now about 100 milliosmoles. And that's because we took a bunch of water out first, making it very salty. And on the way up, we pumped more salt out of it, making it less salty. And that gives us a very powerful way to reclaim not only water, but sodium chloride out of our filtrate. So obviously water is super important, right? Because we need it to maintain our blood volume and our tissue fluid, and dehydration is bad for approximately 1 million reasons. But think about all the times this term we've discussed the importance of sodium or chloride in some kind of exchange. So chloride shift, for example, both in the PCT and in the renal medulla, and in the red blood cells between the alveolus and the tissues. That's another place where chloride is useful. And sodium, we know we use that for all kinds of stuff, notably anything that is doing an action potential, but sodium gradients also power exchange in the PCT and the DCT. So sodium is also a super important ion. So we're getting back two things that we really need using this loop. And then by the time the filtrate reaches the DCT, it's almost finished. It's almost P. We just have to put some finishing touches on it. And the DCT and the collecting duct, which I'll pivot to momentarily, have the capability to continue reclaiming water even after the loop. So a couple things to note about the loop. Since the gradient is set up by urea and this constant pumping, which is not at the mercy of concentration gradient direction because it's active transport. So if we spend some ATP, we can just force things to go where they might not ordinarily be inclined to go. And that sets up this permanent gradient, which powers our water reclamation via salt reclamation. And this is true no matter how hydrated you are. So the loop works the same whether you're dehydrated and in desperate need of water, or whether you're very well hydrated and you don't desperately need the water. So at the renal tubule, we have two kinds or sort of varieties of water reclamation, and they are named according to whether they are obligate, which means must happen, always happens in the same way, or whether they are facultative, meaning you can shift how much water you're reclaiming according to your hydration state and your need. So the loop is obligate water reabsorption. And then the DCT and collecting duct are part of what we call, I'm just going to move my toolbar over here so I can write this. Facultative reabsorption of water, where we can fine tune how much water we're hanging on to or not according to our hydration state. So the reason that's an important distinction is that it really informs your understanding of the role of the kidneys and helps you know that there's a permanent built-in feature of water reclamation that's always going to happen regardless. And then in order to deal with homeostatic needs, we have this adjustable component as well. And so those portions, the adjustable components, switch on with the renin-angiotensin-aldosterone system. So apropos of that, before we pivot over to looking at the juxtaglomerular apparatus and doing a little bit of histology and physiology at the same time, those things I'm going to do in the next video, I'm just going to really quickly run us through the facultative component so that we can compare it to the obligate portion. And before I do that, I'm going to try to take a screenshot of this so that you have a still image to refer to as well as the video. Please work, please work. Save. Actually, I'll just say okay, so now that's saved. So I'm going to erase a couple of things and pivot over to this side of the drawing, but I'm going to leave some of this in place just so that we have an ability to sort of compare them. So I'm just getting rid of my labels so I have room to draw. Okay, so now we have these two additional tubes. So let me just redraw my little direction arrows really quickly. And then I'm just going to include the other DCTs that are plugging into my collecting duct, and then just say the exit is down there. All right, so we've got the same situation with the urea. At the bottom here, where there's a gradient of it. And then let's also add in sodium and chloride, which I'm going to just make the dots for this. Okay, so just like a quick little visual that indicates the gradient. So if I activate the renin-angiotensin-aldosterone system, so what that's going to mean is some special stuff right here that we'll look at in a little bit are going to notice that there's been a change in flow in the kidney and going to switch on the water saving function. So when that happens, we have an increase in sodium potassium pumps in the DCT, specifically both the apical and the basal surface. I'm not going to draw all that here. I'm just going to say, here is our, oh no, I made it the same color as something else. Let's try to avoid that. How about I make it perp? So this is going to be our aldosterone dependent sodium potassium pump. So I'll just label that over here. And there's also sodium potassium pumps that are just permanently present in the distal convoluted tubule. They're not always there. So when you have aldosterone on board, it's not that you're making sodium potassium pumps be in a place they weren't already. It's just that you're adding to the ones that are already there. So that's important to know. So what these are going to do is move sodium back into, excuse me, other way around. Move sodium out of the tubule so we're reabsorbing sodium. And water is going to follow that sodium. So water is like, oh, where are you going? I want to go there too. And I will. I'm just going to follow you. I'm like your annoying little sibling who your mom said you had to take with you if you wanted to go somewhere. That's what water is to sodium. And then this is at the expense of potassium. So we have to exchange the sodium for something. And obviously this is potassium. So what this does is create a scenario where we're getting back sodium and water, which is great, but we're also losing potassium in our urine at a higher rate. And this is why aldosteronism, which is chronic aldosterone secretion, can lead to dangerous hypokalemia, which is the word for low plasma potassium levels. And that starts to mess with your nervous system function and your muscular function because, of course, we need potassium to repolarize excitable cells. So it can be a problem. It's not in the short term. So the other thing that happens when the RAS is turned on is we get antidiuretic hormone inducing the expression of aquaporins. And aquaporins just mean exactly what they sound like. They mean aqua, water, porin, a pore. It's just a water pore. So it's a special channel. Its inner chemistry is such that only water can go through it, not other stuff. But it allows water a way out, both in the DCT and collecting duct. So basically what ADH does is it circulates in the blood from the posterior pituitary down to the kidneys, and it tells the cells of the DCT and the collecting duct to transcribe and translate the protein that is an aquaporin. So it induces gene expression to create extra water holes. And then because we already have this gradient, if holes are available, water will go through them. And it does. So, oops, the heck? Oh. What we end up getting is even more water leaving the urine and being attracted increasingly as it descends the collecting duct, increasingly attracted out because it has a hole to go through. And there's this really strong gradient out here. So water is going to just osmotically be like, ooh, I'm going to be out here. And then it gets put into the peritubular capillaries, the vasa recta, etc. Et voila, we have upped our blood volume. And what we are doing is creating very small volume of very high concentration urine. So if you were to, like, let's say, turn on the renin-angiotensin-aldosterone system in somebody and measure the changes in their urine once you flip it on, you would see a transition from larger volumes of fairly dilute pee to very small volumes of very high concentration urine. So not a whole lot of urine, very dark in color, more odorous than usual, but the key there is not volume. So humans, we have to urinate because we don't have an apparatus to concentrate waste products and reclaim water from them to make like a paste or anything drier. So water loss is a necessary part of waste excretion, which is an unfortunate challenge for us, but we do pretty well at it with what we've got. The reason I say that is that there are other creatures on the earth that don't do this, and instead just make a hyper-concentrated, very nitrogen-rich waste, which comes out as sort of a whitish paste. And creatures that do that include birds. So that's why bird poop has that white part. That's actually their pee. And anything that produces large amounts of guano, also including bats. So if you've ever been to a chicken farm and you know the smell of a chicken farm, which is god awful, that's because of the way that they deal with their ammonia and urea, which is make it into a chalky paste and excrete it along with their feces. We humans have repurposed that into making fertilizer out of it because nitrogen is a limiting nutrient for plants. And then some very mean and nasty people also use that to make explosives because nitrogen is a critical ingredient in that as well. So that's where the idea of fertilizer bombs come from. It's all about different species dealing with nitrogenous waste differently. But we have to lose water to do it. So that's how you limit the water loss. So let me just go ahead and take a screenshot of this. And then I'll end this video and start the next one. Okay.